Dear colleagues, dear friends, thanks to the Congress organizer for this invitation to present a look to basic research and especially to the DINOM research activities and its implication for further research. Today, I will explain what is dynamic pharmacology for homeopathy. The first question we want to address here is, could homeopathy be a placebo only? We prepare two movies to answer this question.
The placebo effect, like the nocebo effect, exists. It acts just as much on the physical body via the brain's neurohormones as it does on the mind. It is present in any medical intervention, even surgery. In a general medical consultation, it may be more marked during a first homeopathic consultation, where the patient's expectation is greatest. This first consultation is the longest and the most detailed. There you have an interaction between patient, physician, and medicine. Compared to an effective treatment, the placebo effect is brief and significantly more variable. A placebo effect, which continues over a period of weeks, is unknown. The duration of treatment is therefore an important parameter to take into account when discussing the question of a placebo effect. In homeopathy, the medicine is taken for only a short period, and so its long-term effects cannot be due to placebo. A placebo effect is relevant only in diseases that may vary, remit, or even disappear spontaneously. It cannot produce curative results in a chronic disease, which by definition is incurable. For example, familial allergic conditions or chronic rheumatic disease. Homeopathy has a good part to play in the treatment of such patients. The placebo-controlled study works best where a drug is systematically given to everyone in a group of patients who all have the same condition. But where treatment is individualized, systematic clinical case studies and patient cohort studies become a much better way of demonstrating the efficacy of homeopathy, and there is no lack of data at this level of evidence. With children, treatment effects should not be compared with placebo, and we can only use comparative studies of clinical cases. Let's play fair. If we're going to insist on studies which include a placebo comparison, we must respect homeopathy's individualized approach. Let's work together without prejudging the outcome. Level 1A of the pyramid can be then filled in, as just shown. This is not always possible or required for other approaches such as psychotherapy, but if it is demanded, let's have the financial means and the necessary conditions to carry out these studies. Let's work together for the well-being of patients. Why do patients use homeopathy? Many pathologies bring patients to a homeopathic doctor. When a prescription is required, 73% of these patients will receive a homeopathic drug. The others will also receive the necessary conventional treatment, mainly in cardiovascular diseases, but also antibiotics. After this first consultation, many patients will be able to stop conventional drugs previously used. This is especially true for tranquilizers and antidepressants. These patients are mostly chronic patients who have suffered from their ailments for an average of more than 6 years. Of course, there is no miracle, and these patients will return as needed for follow-up consultations for their chronic diseases. We also note that the need for frequent recourse to a family doctor before and after homeopathic care is significantly reduced. The same applies to the use of specialist doctors before and after homeopathic care, which is reduced by half. Before they were treated with homeopathy, patients' health is generally very worrying. After the use of homeopathy, although most of the people were afflicted with chronic diseases, physical problems are significantly improved. For psychological problems, we observe exactly the same results. Finally, we can ask about the effectiveness of homeopathic treatments and patient satisfaction. In terms of both physical and mental health, when patients are asked to compare the results obtained by previous treatment and homeopathic treatment, they are clearly in favor of homeopathy. And of course, this results in a different degree of patient satisfaction. For public health, there is no doubt homeopathy is useful for patients. Cost savings are also evident in the demonstrated reduction in the number of conventional prescriptions and consultations. This is without any risk because the essential medicines and the necessary specialist advice are still prescribed by homeopathic doctors. That homeopathy and a conventional approach are complementary is obvious. The reimbursement of consultations and drugs is a very legitimate request from patients.
Because homeopathy is mostly used for chronic disease, as you see, without possible spontaneous healing, it must be more than a placebo. Therefore, next question must be, what is the nature of the homeopathic medication? As you already know, Dinome decided from the start to look at the homeopathic medicine from different points of view: particles, structuration of solvent, and electronic consequences. Here, a movie on what we did and still are doing. Sorry for the quality of the image.
In 2014, the professional body of Belgian homeopathic doctors decided to initiate a research program to address outstanding questions about the nature of homeopathic medicines and the role of the process of potentiation during manufacture. These questions are important to both the prescriber and the patient. Each has an interest in understanding what is being used to treat and cure. It is also important for the pharmacist to know the best methods for manufacturing these medicines. Nowadays, there are very sensitive, accurate, cutting-edge analytical techniques which can provide answers to these questions. From the outset, we decided not to limit ourselves to any single technique, but to look at homeopathic medicines from various angles: their material composition, their impact on the solvent, or their signature, and the electromagnetic image which they produce. We now know that homeopathic medicines, even at the highest potencies, contain small amounts of physical material, which has a definite form and structure. When simple dilutions without potentiation were analyzed in the same way, we found that these no longer contained any specific structure or material. With serially potentiized solutions, the amount of material present stays the same after the fourth centesimal dilution. Pure potentiized water contains no specific material. When a solution is potentiized, the water solvent takes on a new structure, which has a pattern derived from the starting substance. Such water therefore holds information, carried in the form of a specific signature, something which has been confirmed by data from our nuclear magnetic resonance experiments. In practice, however, pharmaceutical prescriptions are manufactured and delivered in various forms, such as granules, pills, tablets, or water alcohol drops. Whilst we were able to show that liquid homeopathic medicines, each have their own specific structures, we wanted to know if this information is carried through into granules or other pharmaceutical preparations. And if it is, does it remain stable as something that living organisms can recognize? We used electrophotonic analysis to make this signal visible. Stimulate a pellet or a drop of water alcohol solution electrically, and you can see it emitting light. In this way, we can visualize the information present in the remedy. The shape and brightness of the emission is specific to each remedy and can differentiate them. It is even possible to discriminate between different potencies of the same medicine. Preparations which are more than 20 years old still retain this information. As soon as it comes into contact with a living organism, it is released and produces a reaction. Naturally, these results will have to be replicated and verified in the years to come. They are nevertheless consistent with results obtained elsewhere, such as in epigenetics, and also with data from traditional homeopathic research, such as provings.
Dinom already published 2 papers, one on the materiality of homeopathic medicine and one on the solvent behavior. Now we will try step by step to understand these results and put them in perspective of the homeopathic practice, proving, and materia medica. First of all, the materiality of homeopathic medicine. If particles are detected, will dynamic pharmacology be able to control these particles? A little movie will show how important this question is. Particles in homeopathic solution were identified using the nano-tracking analysis device. Therefore, the liquid of homeopathic medicine is passing before a laser beam, illumining them, allowing counting and measures of their size. Here, some examples of what the laser beam sees. That is cuprum metallicum simply diluted at a level of 10⁻⁸. Then you have centrum for centesimal Hahnemannian dilution, dynamization. Then you have cuprum 30 centesimal Hahnemannian dilution, dynamization. And cuprum 200 Korsakovian dynamization. Such counting has been reproduced several times, 5 or at least 3 times, allowing statistical analysis later on. The size of particles are the widest, but showing also more diversity for the 30 centesimal Hahnemannian dynamization. Here are some examples of particles measured comparing cuprum metallicum particles with gelsémium sempervirens particles. Of course, we used also several controls, including dynamized water and other medicines prepared in glass or polyethylene containers. Size and number of particles are indeed controlled by the dynamization process and only by the dynamization process. We have particles specific in size and number. We know that now. But does the dynamic pharmacology also control their shape? Therefore, the scanner electron microscopy can be used. The particle shapes are specific for a manufacturing line of a stock only when a dynamization process has been applied. Here above, some of the observer shapes for cuprum metallicum compared to gelsémium sempervirens under in this side. They are really totally different. They are controlled. Next step is to analyze the chemical composition of this material. This can be observed with a semi-quantitative device, the energy dispersive X-ray spectroscopy. A drawing program allows to elaborate models of these particles. All we need is a proportion of chemical compounds in each particle. The size of the real detected particles are at least 200 times bigger than the one presented here, but in the same chemical composition. You see here carboxylicate skeleton expressing at the outside electric charge ions. As a 6 decimal or 3 centesimal dynamization, we still have traces of source material. All this has been mathematically verified and published. Of course, verification and confirmation are needed in the future. Okay, but does dynamic pharmacology also have an impact of water solvent? To explain nuclear magnetic resonance, we have to focus on the motion of hydrogen nucleus in water molecules. Each nucleus is turning on itself, producing a little magnetic spin. If we apply an external magnetic field on this system, the nucleus spin of hydrogen will align themselves on the external spin. This phenomenon takes some time, and we can calculate the relaxation times needed to obtain the new equilibrium. This calculation may be done on the vertical and horizontal axis T1, T2. If water is not pure, containing some particles, this water will be organized around these particles, and the relaxation times will depend on this fact. As such, you get an indirect view on what interferes on the hydrogen molecule nucleus, allowing even nuclear magnetic resonance imaging, because water and hydrogen nucleus are the most important constituent of the body. Doing the measurement of this relaxation time for homeopathic solution, we obtain typical ups and down Kolikoff-Steiner curves for full manufacturing line. Here we have a full manufacturing line of Hahnemann dynamization from 4CH to 30CH of cuprum. After our first publication on this topic, we had to verify these results. We reproduced the same measurements on 2 supplementary manufacturing lines of the same medicines and same controls. Confidence intervals are very close for T1 relaxation. Here's the example of cuprum presenting an ascending linearity. Here you have the gelsémium sempervirens presenting a descending linearity. And here you have a lactose dynamized control. Confidence intervals are not as close as the previous one. No ascending or no descending linearity. Here you see the statistical result of pooled results from the 3 manufacturing line. The statistical discriminance between cuprum and gelsemium is confirmed. But we found also a statistical significant variability of measurements between manufacturing line of the same preparation. The reason for this discovery must be found in some covariant that partially hides the homeopathic signature in the solution. Probably the most critical factor is the quantity of free oxygen in the sample. This quantity is linked to the temperature, humidity, atmospheric pressure, and other external factors during manufacturing process. Measuring the concentration of free oxygen in the nuclear magnetic resonance sample, we could confirm that the free oxygen is indeed a covariant. Here a comparison of the free oxygen concentration, line above, and the T1, T2 relaxation time. This example of cuprum metallicum centesimal anomanian manufacturing line is self-explanatory. At 4C, the oxygen concentration is very low, without influence on the relaxation time. At this concentration, we still have a lot of lactose, 9,500 micrograms per gram of solution. And lactose is eager for oxygen and absorbs it very quickly. At 21C, we found a clearly lower concentration of oxygen, and this time, the relaxation time is clearly influenced by this fact. For gelsemium, the same phenomenon can be observed at 3C. Alkaloid markers of the plant are still part of this preparation. Further, in this manufacturing line, we found no link between free oxygen concentration and relaxation time. Dynamic pharmacology of specific source material, cuprum or gelsemium here, is the reason for longer relaxation time compared with all their contours. The dynamization process of a mother tincture or stock induce a specific organization in the solvent. These facts are now confirmed and verified. We hope for a publication of these results as soon as possible. Okay, we have measured physical particles. At the level of hydrogen nucleus, we have measured change in the solvent. But what about the electrons in the solution, or even in the homeopathic globules, pills, or granules? To obtain some insight on this question, we will use electrophotonic imaging. It's a very new method. We are in the process of validation of this method. Here, some theoretical explanation of this technique.
Here's a very simplified model of an atom. The nucleus at the center of the atom is where the protons and neutrons live. But they're kinda boring because for the most part they just sit there. The real star of the show is the electron. The electron gets to do all the interesting stuff like move around, jump around, bind with other atoms. These dashed lines represent the different energy levels the electron can have while in the atom. We like representing these energy levels with an energy level diagram. The energy level diagram gives us a way to show what energy the electron has without having to draw an atom with a bunch of circles all the time. Note that the electron for our hypothetical atom here can only exist with 0 eV, 4, 6, or 7 eV. The electron just cannot exist between energy levels. It's always got to be right on one of the energy levels. Okay, so let's say our electron starts off on the 0 eV energy level. It's good to note that the lowest energy level an electron can have in an atom is called the ground state. So how could our electron get from the ground state to any of the higher energy levels? Well, for the electron to get to a higher energy level, we've got to give the electron more energy. And we know how to give an electron more energy, you just shoot light at it. If a photon of the right energy can strike an electron, the electron will absorb all the photon's energy and jump to a higher energy level. The electron in this ground state needs 4 eV to jump to the next energy level. That means if a photon that had an energy of 4 eV came in and struck the electron would absorb the energy of the photon, causing the photon to disappear, and that electron would jump up to the next energy level. We call the first energy level after the ground state the first excited state levels. What if our electron's in the ground state and we send a 5 eV photon at it? If the electron were to absorb all of the energy of the 5 eV photon, it would now have 5 electron volts. But that's not an allowed energy level, so the electron can't absorb this photon, and the photon will pass straight through the atom. Keep in mind the electron in the atom has to absorb all of the photon's energy or none of it. It can't just absorb part of it. Alright, you could also ask about the emission spectrum. The emission spectrum are all of the wavelengths or energies that an atom will emit due to electrons falling down in energy levels. You could go through all the possibilities of an electron falling down again, but you'd realize you're going to get the exact same energies for the emission spectrum that you got for the absorption spectrum. So instead of letting light pass through a gas composed of your hypothetical atoms, let's say you made a container that had the gas of your hypothetical atoms and you ran an electric current through it, exciting those electrons to higher energy levels and letting them fall back down to lower energy levels. This is what happens in neon lights. Or if you're in science class, it's what happens in gas discharge tubes. So for the emission spectrum, instead of seeing the whole electromagnetic spectrum with a few lines missing, you're going to only see a handful of lines that correspond to the energies of those photons that atom will emit.
The user's device can be formatted to observe the image produced after electrical stimulation around 1 drop of a hydroalcoholic solution or 1 globule. The camera must be refrigerated at min 60 degrees centigrade Celsius to be able to observe the emitted light, because 80% of this light is in the UV spectrum. 1 drop or 1 globule is emitting light in a specific way, a corona of ray for a drop, and concentric rings for a globule. For a globule, the primary control are the neutral one, and the one impregnated with pure solvent. The distribution of the wavelengths of the emitted light is here of a very simple shape and clearly circular, as you can see. Everything changes when the globules are impregnated with homeopathic medicine. The wavelength structure of emitted light, or entropy, allows a discrimination between remedies, cuprum, germanium, but also between dynamization levels and between dynamization methodologies such as Korsakoff preparation. Using this methodology, a clear discrimination between impregnated globules with full line of centesimal Hahnemannian potentization of germanium and cuprum is clearly possible. You see the 2 lines here. There are no one of the measurements that is matching each other. Here we are measuring the number of pixels, this of light, a pixel emitted in the selected area. This area includes the numerously most represented large wavelengths of the emitted light. It is the same for Korsakov preparation. Here we are measuring the orientation angle of the selected area. Like you have seen, they make a form and we have an angle. Here we are comparing all measurements sought out in ascending values for pure globule, it's a line above, with solvent impregnated globule, just under, centesimal Hahnemannian lines for germanium and cuprum, and Korsakoff lines. Homeopathic globules are not empty or solvent impregnated globules. It's something different. The mathematical and statistical analysis of the collected data is ongoing at the University of Strasbourg, considering also full lines of simply diluted control. Globules results are prioritized for publication, but later on, the same will be done for liquid drops. Don't say that you cannot see here the difference between 1 drop of cuprum 50,000 Ki and 1 drop of gelsemium 50,000 Ki. Korsakoff dynamization. Here are some preliminary statistical results from the analysis of the mean value of several structural parameters from global, circumference, area surface, angle, and so on. Comparing cuprum images with gelsemium images and with their own simply diluted controls, p-values are all significant. For drops, we anticipate also discriminant value for light contrast and light energy. This is not yet published, but in working process for 2 separate publications. We foresee already some conclusion, as already explained, but analyzing also old sample, some of 30 years old, we could see that the discrimination between remedies stays stable over time and seems even improved in energy and contrast. This is what we know from all this measurement. Homeopathic medicine are not empty. Structural changes in the solvent are a consequence of the dynamization process. The specific information remains in solution and pills. Dynamic pharmacology is more than a dilution process. To explain dynamic pharmacology, we have to consider the coexistence of particles and waves. Particles are physical particles, solvent molecules, nucleus, and electron. Waves are of electromagnetic nature, electrons or photons. But in between, we have to look also at the predicted, by new physics, the predicted excitons. It is a mix of material and light in such system. The concerned research fields are chemical identification, electromagnetic detection. Explanation will come from physics and quantum mechanics. Homeopathic medicine is a very complex system. So far, Dinam investigated the presence of particles, solvent behavior, and electrophotonic information in the homeopathic medicines. These aspects of dynamic pharmacology have to be investigated further, and other questions need answer. As example, how the homeopathic medicine influence health of patients or produce proving symptoms. Modern scientific developments are open doors to these questions. But this question is in fact already answered. We need epigenetic to understand it.
Here's a conundrum. Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with the multitude of smaller molecules found within cells, which can activate and deactivate genes. If you think of DNA as a recipe book, those molecules are largely what determine what gets cooked when. They aren't making any conscious choices themselves. Rather, their presence and concentration within cells makes the difference. So how does that work? Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means that they could affect an organism for its entire life. Sometimes that's a good thing. Epigenetic changes are part of normal development. The cells in an embryo start with one master genome. As the cells divide, some genes are activated and others inhibited. Over time, through this epigenetic reprogramming, some cells develop into heart cells and others into liver cells. Each of the approximately 200 cell types in your body has essentially the same genome, but its own distinct epigenome. The epigenome also mediates a lifelong dialogue between genes and the environment. The chemical tags that turn genes on and off can be influenced by factors including diet, chemical exposure, and medication. The resulting epigenetic changes can eventually lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason why genetically identical twins can grow up to have very different lives. As twins get older, their epigenomes diverge, affecting the way they age and their susceptibility to disease. Even social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their pups, genes in the babies that help them manage stress were methylated and turned off. And it might not stop with that generation. Most epigenetic marks are erased when egg and sperm cells are formed. But now, researchers think that some of those imprints survive, passing those epigenetic traits on to the next generation. Your mother's or your father's experiences as a child or choices as adults could actually shape your own epigenome. But even though epigenetic changes are sticky, they're not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may, in the long run, create a healthy epigenome. It's an exciting time to be studying this. Scientists are just beginning to understand how epigenetics could explain mechanisms of human development and aging, as well as the origins of cancer, heart disease, mental illness, addiction, and many other conditions. Meanwhile, new genome editing techniques are making it much easier to identify which epigenetic changes really matter for health and disease. Once we understand how our epigenome influences us, we might be able to influence it too.
The dynamic homeopathic medicine is a complex system, as we saw, but you see that complexity is also the norm in any living being. These steps are needed to explain how dynamic pharmacology can be converted into informational medicine. About epigenetic, Professor Bellavete and others have already published key knowledge about more than 20 homeopathic medicines. We know, as example, that Gelsemium sempervirens significantly upregulated 7 genes and downregulate 49 genes of the 22,000 genes we have. The 7 stimulated genes are expressed in the heat shock protein system, and the 49 inhibited genes are all expressed in the neuron lowering anxiety. As explained by Dr. Ashley Ross at the previous Liga Medicorum Homeopathica Internationalis Congress in Cape Town, the validation of homeopathic proving and/or repertory symptoms is difficult. An answer to this problem can be obtained making a link between the activated or slowed down genes observed by epigenetics and the observer's symptom from a proving and clinical practice. By the validation of their symptoms, a link between traditional experimentation in homeopathy and modern science can be achieved. Homeopathy is modern and innovative science. Homeopathy has been built on a long tradition, and new research confirms the visionary view of Hippocrates and Hahnemann. Dynamic pharmacology, materiality, and complexity needs to be investigated further for the well-being of humanity. In Europe, we have a legal frame for the registration of homeopathic medicine. Basic research is indeed needed to maintain availability of homeopathic medicine on the market in future.
Here's a conundrum. Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with the multitude of smaller molecules found within cells, which can activate and deactivate genes. If you think of DNA as a recipe book, those molecules are largely what determine what gets cooked when. They aren't making any conscious choices themselves. Rather, their presence and concentration within cells makes the difference. So how does that work? Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means that they could affect an organism for its entire life. Sometimes that's a good thing. Epigenetic changes are part of normal development. The cells in an embryo start with one master genome. As the cells divide, some genes are activated and others inhibited. Over time, through this epigenetic reprogramming, some cells develop into heart cells and others into liver cells. Each of the approximately 200 cell types in your body has essentially the same genome, but its own distinct epigenome. The epigenome also mediates a lifelong dialogue between genes and the environment. The chemical tags that turn genes on and off can be influenced by factors including diet, chemical exposure, and medication. The resulting epigenetic changes can eventually lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason why genetically identical twins can grow up to have very different lives. As twins get older, their epigenomes diverge, affecting the way they age and their susceptibility to disease. Even social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their pups, genes in the babies that help them manage stress were methylated and turned off. And it might not stop with that generation. Most epigenetic marks are erased when egg and sperm cells are formed. But now, researchers think that some of those imprints survive, passing those epigenetic traits on to the next generation. Your mother's or your father's experiences as a child or choices as adults could actually shape your own epigenome. But even though epigenetic changes are sticky, they're not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may, in the long run, create a healthy epigenome. It's an exciting time to be studying this. Scientists are just beginning to understand how epigenetics could explain mechanisms of human development and aging, as well as the origins of cancer, heart disease, mental illness, addiction, and many other conditions. Meanwhile, new genome editing techniques are making it much easier to identify which epigenetic changes really matter for health and disease. Once we understand how our epigenome influences us, we might be able to influence it too.
In Europe, 2 directives require the registration of homeopathic medicines for human and veterinary use without therapeutic indication. We are talking about a simplified registration procedure. In national procedure, however, an indication can be authorized on the basis of the evidence of effectiveness submitted for registration. This is referred to as a full registration procedure. Some homeopathic medicines are reserved for medical prescription because of this indication. In European procedure, it is necessary for the consumer to be able to know the following: the exact name of the strain and its degree of dynamization, the registered office of the owner of the registration, the mode and route of administration, the expiry date, the capacity, the batch number of the manufacturing batch, the apparent mention, homeopathic medicinal product without specific therapeutic indications, and a warning advising the user to consult a doctor if symptoms persist. A registration file consists of 5 parts: administrative, including the applicant's manufacturing authorization certificates, laboratory quality control, qualitative, with all the procedures for strain identification, stability, validated in-process control procedures, toxicological, to ensure the safety of homeopathic medicines, we check whether what will be delivered to the patient is indeed in a safe dilution, even for a newborn who swallows a whole vial. The alcoholic content will of course also be checked. Homeopathic use, based on traditional, clinical, and other publications justifying such use. Concise summary of all these controlled data. Even if marketing authorizations are national, a harmonization of registration procedures is controlled by a European working group, the Homeopathic Medicinal Products Working Group, under the supervision of the heads of European medicines agencies. Head of Medicine Agency. Production procedures are also controlled to produce the desired dynamic changes. For example, DH or CH or K. The homeopathic pharmacist, preparer, uses certified stocks. His laboratory must meet European standards. This registration is the ultimate guarantee of the quality, safety, and proper use of homeopathic medicines for and by patients.
But we have still many things to do. This is only the very beginning. We need confirmation, we need verification, we need broadening to other source material, looking to different manufacturing techniques, and the impact of this new knowledge for practice. More research teams must be put in place, and in parallel, the theoretical frame behind all these results must be developed carefully. Thank you for your attention. Maybe you have questions, it will come at the end of the phone.